Chapter 26, the urinary system. Page 1. Overview. A. Functions. Excretion of wastes from fluids. Elimination of wastes to environment. Homeostasis of plasma volume and solute concentration. Components. A. Kidneys. Blood vessels and nerves enter and exit at the hilum, which is an indentation. The kidneys filter waste from blood. The renal capsule, which is a fibrous capsule, and adipose capsule protect the kidneys. Renal fascia anchors the kidneys. 1. Kidney section. A. Cortex. This is the superficial region that contacts the capsule. Renal columns within the cortex are deeper extensions of the cortex. B. Medulla. This is deep to the cortex. Within the medulla, the renal pyramids are triangular structures with a base at the cortex. The renal lobes within the medulla contain nephrons, and this is where urine production occurs. C. Pelvis. This is the funnel-shaped chamber in the center region of the kidney. Within the pelvis are the minor calyx funnels urine from the nephron into the major calyx. The major calyx funnels urine into the ureter. B. Ureter. This is a muscular tube from the kidney to the bladder. C. Urinary bladder. This is a muscular organ with rugae that stores urine. Page 2. D. Urethra. This is a tube from the bladder to the exterior. Nephrons. These are tubular structures that produce urine. A. Anatomy. 1. Renal corpuscle. This produces filtrate, also known as blood plasma. A. Glomerular capsule, also known as Bowman's capsule. This is a cup that insulates capillaries and captures filtrate. The glomerulus is a fenestrated capillary network where fluid is forced from blood into capsule. The afferent arteriole carries blood to the glomerulus. The efferent arteriole carries blood from the glomerulus into peritubular capillaries. B. Filtration apparatus. The fenestrated capillaries are porous vessels that let fluids and some solutes pass. The lamina densa is a dense layer. It contains lots of glycoproteins, which are a web-like meshy filter. It restricts larger solutes and keeps them in the blood. Filtration slits. These are gaps created by foot-like extensions called pedicles from podocytes that cover the capillary wall. 2. Renal tubule. This functions in reabsorption and secretion. A. Proximal convoluted tubule. This reabsorbs water and ions from tubular fluid into peritubular fluid, or interstitial fluid. It recaptures smaller solutes from what has already been filtered. B. The loop of Henle, also known as the renal loop. This reabsorbs water, sodium, and chloride from the tubular fluid and sends it to the peritubular fluid, which is interstitial fluid. The loop of Henle has thick and thin, ascending and descending limbs. Page 3. C. The distal convoluted tubule, also known as the DCT. This secretes ions, acids, drugs, and toxins from the peritubular fluid to the tubular fluid because of the large size of the objects being secreted. Reabsorption occurs in the DCT. Reabsorption of water, sodium, and chloride from the tubular fluid to the peritubular fluid. Juxtamedullary complex. This is made of macula densa, which are DCT cells near the renal corpuscle, and juxtamedullary cells, which are smooth muscles of afferent arteriole. The juxtamedullary complex secretes renin for angiotensinogen conversion, as well as erythropoietin for red blood cell formation. 3. The collecting system. This secretes and reabsorbs water, sodium, potassium, hydrogen ions, and bicarbonate HCO3 negative. The fluid moves to the papillary duct. B. Types of nephrons. 1. Cortical nephrons. These are found in the cortex and in the short loop of Henle. They perform the most reabsorption and secretion. The cortical nephrons are efferent bodies sending material to the peritubular capillaries. 2. Juxtamedullary nephrons. These are found within the medulla and the long loop of Henle. They are important for water conservation and the formation of concentrated urine. Juxtamedullary nephrons are efferent bodies moving material from peritubular capillaries to the vasa recta, which are capillaries that parallel the nephron loop. Urine. A. Filtration in the glomerulus. Water and solutes are forced from blood into the capsular space. B. Reabsorption in renal tubule. This occurs within the proximal section. Water and solutes move from tubular fluid to peritubular fluid and then into the bloodstream. C. Secretion in the renal tubule. This occurs within the distal portions. Solutes move from peritubular fluid to tubular fluid. Filtration. A. Pressures. Glomerular hydrostatic pressure. This is the strongest pressure. Blood pressure within the glomerulus pushes water and solutes from plasma to filtrate. 2. Capsular hydrostatic pressure. This is the resistance of nephrons that pushes water and solutes from filtrate to plasma. It opposes glomerular hydrostatic pressure. Page 4. 3. Blood colloid osmotic pressure. This is the osmotic pressure of protein that pulls water and solutes from the filtrate into the plasma. It opposes glomerular hydrostatic pressure. 4. Filtration pressure, abbreviated FP. This is the average pressure that forces water and solutes from glomerulus to capsular space. The equation is GHP minus CHP minus BCOP. B. Glomerular filtration rate, abbreviated GFR. This is the amount of filtrate produced per minute. A change in filtration pressure affects GFR. Control of GFR. A. Autoregulation. A change in afferent and efferent vessel diameters. If GHP and GFR go down, then afferent diameter goes up. Efferent diameter goes down. This will lead to an increase in GHP and GFR. The same works in reverse. B. Hormonal regulation. If GHP and GFR go down, then angiotensin II causes efferent diameter to decrease. This will lead to an increase in GHP and GFR. C. Autoregulation. If GHP and GFR increase, then the sympathetic nervous system causes afferent diameter to decrease. This will lead to a decrease in GHP and a decrease in GFR. Reabsorption. A. Reabsorption at the PCT, proximal convoluted tubule. 60 to 70% of water, ions, and organic nutrients are absorbed here in the PCT. B. Reabsorption at the loop of Henle. Water, sodium, and chloride are reabsorbed via countercurrent multiplication, abbreviated CM. CM is exchanged between fluids moving in opposite directions. The thick ascending limb of the loop of Henle. This is impenetrable to water. The thick ascending limb is responsible for the active transport of sodium and chloride from tubular fluid to peritubular fluid. It creates an increase in osmotic concentration in the peritubular fluid around the thin descending limb. The thin descending limb. This is impenetrable to solutes. Water diffuses into peritubular fluid toward an increase in osmotic concentration. Benefits of CM. Efficiency in the reabsorption of water and solutes before tubular filtrate reaches DCT. ADH increases reabsorption from the collecting duct into the bloodstream. Page 5. C. Reabsorption and secretion at the DCT. Water, sodium, and calcium are reabsorbed. Aldosterone-controlled pumps reabsorb sodium and secrete potassium. Potassium, hydrogen, and ammonia are secreted. D. Reabsorption and secretion at the collecting system. Water, sodium, bicarbonate HCO3 negative, and urine are reabsorbed. Aldosterone-controlled pumps reabsorb sodium and secrete potassium. If paratubular pH decreases, then hydrogen ions will be secreted and bicarbonate will be reabsorbed. If paratubular pH increases, then hydrogen ions will be reabsorbed and bicarbonate will be secreted. E. Summary of renal function. Urine volume and osmotic concentration. A. Obligatory water reabsorption. This is when water reabsorption cannot be stopped. It is due to paratubular osmotic concentration. This occurs in the PCT and the descending run. B. Facultative water reabsorption. This water reabsorption is controlled. It is controlled by antidiuretic hormone. It occurs in the DCT and collecting system. Micturition. This means urination. A. Process. As the bladder fills, there is an increase in neural activation and fluid pressure. External and internal sphincters relax. Aging in the urinary system. Aging is associated with an inability to control micturition. Nephron function decreases, and sensitivity to ADH also decreases.